Hi guys, this is a video to go over your end of topic homeostasis test to make sure that you're fully confident with the answers. We're going to do a bit of exam technique and then we're going to go over the specific things that you needed to do to hit the marking points. So first of all, exam technique, when you're reading a question, make sure that you're highlighting or underlining as you go to slow your brain down and focus you on what the question's asking you, and then do a bit of annotation or just a brain dump above or below at the side of the question to really free up your mind so that when you are focusing on the questions and trying to analyse those later in the question, you don't have to be thinking about recalling the knowledge that you're probably going to need for it as much. You can focus on what is the question asking you. So here it's to do with blood glucose, so I've put insulin and glucagon are your two hormones that control blood glucose. Insulin is released from the pancreas when you've got too much sugar in your blood. Glucagon is the hormone that's released when you have not got enough glucose in your blood. It's then showing you that we're going to be focusing on if there's a decrease in the concentration of glucose in your blood. For example, if you haven't eaten for a long time, that's going to be detected. A hormone's going to be secreted. So I've already thought about what that is. And then we're going to be breaking down glycogen, which I've put down is glycogenolysis, and that's in the liver to be able to release glucose to increase the concentration back to the normal level. So we've thought about it already, got our heads around our knowledge before we're then going on to freeing up our brain to analyse the question. So name the hormone, that's glucagon, and it must, not great handwriting there, that's an O, it must be spelt correctly. If it's not spelt correctly, in this case, it could be confused with another word and the examiner would not give you the mark. So for example, if you put a Y there by accident, they might think you don't know whether it's glycogen or what it is. So glucagon is the hormone, and if spelling's not your thing, just writing it out lots of times, reading it lots of times and testing yourself on it is the way forward. The organ in which glycogen is stored is the liver. So if you didn't know these two, these are GCSE knowledge. That's not to kind of berate you if you got them wrong. It just means you perhaps haven't revised this topic and you haven't had enough time to revise it little and often at home yet. And we need to be focusing on keywords in each topic and knowing what they are. So maybe making flashcards, testing yourself little and often, or asking someone to get involved to help test you. For example, each other would be good. Next question, explain, so that's give reasons why this example is described as negative feedback. All of you know the idea of negative feedback. Not all of you described it in a way to get the marks. So what I would advise you do, I know this is a bit of a beast of a sentence, but have a sentence which includes all of these points that I'm going to go through, and then just be able to revise it so much that you can regurgitate it. So test yourself on whether you can remember it later today. Test yourself tomorrow. Test yourself in a week. Test yourself in a month, and then it will be there ready in your long-term memory to retrieve as you need to in the exam. So negative feedback is when there's some sort of change away from the normal line, and that could be an increase or a decrease. And when the body detects that change, there is a response produced which will reverse the change that occurred in the first place. So the key bits are highlighted there for you in green. So in this example, we've got a decrease in glucose concentration being detected, a hormone secreted, and then we've got a response created, which is going to, instead of further decreasing, the change was a decrease, that would be positive feedback if your body did something to decrease it further. Negative feedback is doing the opposite of decreasing, which is increasing to get back to that normal level. If we had an increase in something, so this is obviously blood glucose, but it could be water potential in the blood, it could be temperature of the body. If we had an increase away from the normal level of anything, then something would occur in the body to be able to produce the opposite effect. So if we had an increase, the response would be decreasing levels to get back to the normal level of whatever we're talking about in homeostasis. Okay, question C. We needed to use knowledge from year 12, biological molecules from Mr. Goodwin. I'm so sorry for how this is probably making you feel seasick. My phone has given up on me and I'm trying to balance my iPad in one hand. So I'm really sorry about the wobbliness of this. So glycogen, we have to talk about the structure and relate it to its function in maintaining blood glucose. So glycogen is branched, is 1 mark, and that is useful because it gives lots of ends for condensation reactions or hydrolysis, depending on whether we want to be releasing glucose into the blood or taking glucose out of the blood and storing it as glycogen. So that would be 2 marks. An alternative 2 marks would be a structural feature is that glycogen is a polymer and it's made up of alpha glucose monomers, and that means that it can release glucose when required to increase the concentration in the blood. A bit later on I did a little bit of a scribble of not the best version of glycogen I've ever drawn, I'm sure, but just to give you the idea that you would have many of these branches. We've got the monomers glucose joined together by, do you remember the type of bond? Yep, well done, glycosidic bonds joining the glucose together. So another marking point for the feature of the structure would be 1 mark for saying glycosidic bonds bond together the glucose in the polymer, and the second mark would be for saying something like these can be easily broken in order to, or easily made to store glycogen, and also easily broken to release glucose into the blood when needed. So that was question C. Question 2, osmoregulation. So we're talking about nephrons in the kidney and making urine. Osmoregulation is controlling the water potential of the blood. I was quite generous. If you put controlling the water levels, I gave you the mark, but please next time be more specific and put water potential, because that's what it is. This next question, so in one of my outtakes of this video, a beautiful diagram, as you can see here, really clear what that is, just to help us talk through this table. So people got a little bit confused with this, so let's just have a look before we look at the question, which is filling in these numbers. The answers are 0.0 for all of them to get 2 out of 2. If it was 0 that you put for all of them, that's the correct thinking, but you got 1 mark out of 2 because we have to record values to the same number of decimal places. So they themselves have done it wrong here by scientific convention in order to make a good results table. So in past exam questions, they do sometimes like asking you what could be improved with this results table, and it might be like another improvement here would be they haven't put the units in brackets or separated by a slash in the heading here. So 0.0 for all of them because you need to record to the same number of decimal places, and 0 as in the data should be 0 for nothing for these because of the following explanation. So protein, glucose and urea are found in your blood. We know that the first step in the nephron in the kidneys is ultrafiltration, and any molecules, so this is the glomerulus here guys, just imagine with all your might, the glomerulus sitting in the Bowman's capsule, we know the blood is under very high hydrostatic pressure because the afferent arteriole bringing blood in is under high pressure, and the efferent arteriole has got a smaller lumen, so it's creating this high pressure and it will force anything small enough out. Protein is too big. Protein remains in the blood. Protein molecules are large, they do not get forced out. So there's none of them in the Bowman's capsule, there's none of them in the PCT, proximal convoluted tubule, there's none of them in the loop of Henle, collecting duct or your wee or anywhere. So 0 for protein because of that. Glucose, however, is small enough and it does get forced out of the glomerular capillaries into the Bowman's capsule. However, we need to reabsorb 100% of that glucose. None of it comes out in your urine. All of it needs to go back into our blood. And we know that in this scribble here, which is the proximal convoluted tubule, this is where reabsorption of glucose happens by co-transport. And the key word here is end. So if it didn't say that, if we were talking about is the glucose here at the start or the middle of the proximal convoluted tubule, the answer is yes, there would be some glucose because it hasn't probably all been reabsorbed back into the blood capillaries surrounding it yet. However, I couldn't guess the number of the concentration of glucose because it would depend on the person and how much they'd had to eat, etc., as to how much glucose was in the blood at the time. So the key word there is the filtrate at the end of the PCT, so right about here before we go into the loop of Henle, there wouldn't be any because all of it has been reabsorbed back into the blood as long as you are a healthy non-diabetic person. So if all of it has been reabsorbed back into the blood, there's none of it here, and then there'll be none of it to be able to create the urine later. Okay, if you turn over for me, onto the next page. Okay, loop of Henle, everybody got that? Awesome, well done, good knowledge, guys. Onto this one, the gradient of ions leads to concentrated urine production. Explain how. If you didn't talk about ions, the question's asking you to talk about ions, so read the question more carefully, go back to your exam technique of highlighting, underlining, circling, whatever key bits in the question. So I'm going to talk you through the main idea, and then we'll look at what I've written and what specific things got you the marks. So the main idea is we want to get, if we're getting concentrated urine because we haven't had very much to drink in our diet, and we want to get a lot of water reabsorbed back into the blood, so there's less water being excreted in urine, then what happens is we want to try and get water moved. This is the collecting duct, this is the loop of Henle. We want water to move out by osmosis. So ADH is involved, and we'll talk about that later in the test. But to get water out of anywhere into another area, think about what has to happen. How does water move in an organism? Water moves by osmosis. What is osmosis? Water moving from high to low water potential. So we want to make sure that the water potential outside of the collecting duct, and this area is in the medulla of the kidney, it's surrounding the nephron tubules. We want to make sure the this area around the outside of the nephron has got a low water potential, so water can move out from high to low water potential. And the way we do that is maintain a high concentration of ions, because if we have a high concentration of ions in the tissue fluid in the medulla here around the loop of Henle and the collecting duct, high concentration of solutes, which are ions in this case, means that there's less free water available, so that's an area of low water potential, and water is likely to move from high to low water potential by osmosis. Little side note, do not put osmosis. I don't know whether another teacher has told you that, but it's not a word in biology. We do say diffuses, don't we, if we're talking about diffusion, but osmosis isn't a word. Say moves by osmosis. Water molecules move by osmosis. Okay, so the marks were specifically for, I gave a benefit of the doubt one, or sort of an extra marking point, because I think it was useful to say this first bit first, and then these ones were the marks. You could get a maximum of 3, obviously. So I would give a mark for saying that sodium ions and chloride ions are diffuse out and are also actively transported out of the ascending limb of the loop of Henle here, and go into the tissue fluid surrounding it in the medulla. So on the mark scheme, you got a mark for saying that ions go into, diffuse or are actively transported into the tissue fluid around the loop of Henle and collecting duct. In that happening, it lowers the water potential there. That was a mark. I'll put that because I was running out of room, but I would write it out in full in your exams, please. And this makes water leave the filtrate by osmosis. 1 mark for saying that water is coming out of the nephron, and 1 mark for saying that the way it comes out is by osmosis. So that is definitely one to revise. Make some little bullet points, a flashcard, however you make your notes, of this is the countercurrent multiplier and how the loop of Henle works. Ions are transported out of it so that it lowers the water potential surrounding the nephron, so that water moves out from high to low water potential by osmosis. That is it. Okay, the bottom one, bit of maths. We had to work out the increase in volume of urine produced in the second 30 minutes. So here's the second 30 minutes between here and here, between 6, so 0 to 30 is obviously the first 30. This is the second 30 seconds. So to calculate, first of all, the volume of urine produced in that second 30 seconds, we need to do 246 take away 81. We then need to know that as a it says it compared with the volume produced in the first 30 minutes, which was 81. So to work that out, that change in, that amount or volume, of urine in the second lot of 30 minutes, we work it out as a percentage of 81 by doing divide by 81 times 100 to give you 204%.